Good evening, everybody. My name is Leila Peivasteh Roudsar from Food and Drug Administration of Iran. I'm in charge of evaluating the safety of imported edible oils and oilseeds. I'm very pleased and honored to have the opportunity to have a part in this important congress and to share my recent research on mycotoxins. In this presentation, I'm going to talk about mycotoxins, their impact on health, and the strategies for prevention and detoxification in food and feed. I have divided my presentation into two parts. First, I'm going to introduce mycotoxins and their adverse effects on human and animal health. I plan to be brief. Mycotoxins are secondary toxic metabolites produced by fungi. They are carcinogenic, mutagenic, teratogenic, and hepatotoxic. So far, liver, renal, and neural disorders, suppression of the immune systems in human and animal through ingestion and inhalation have been reported. Diseases caused by the intake of mycotoxins are known as mycotoxicosis, which doesn't necessarily require the presence of fungi after the mycotoxin formation. The presence of these toxins not only affects the health of consumers but also has an impact on global trade. Currently, more than 300 types of mycotoxins have been recognized. The term mycotoxin was first introduced in 1962 after a crisis in London known as Turkey X disease, which led to the death of 100,000 turkeys. The consumption of peanuts contaminated with secondary metabolites of Aspergillus flavus was diagnosed as the main cause of the disease. This event has drawn scientists' attention to the toxic and fatal metabolites in another fungus. Some of the mycotoxins with the highest impact on human health and economic status include aflatoxins, ochratoxins, trichotisms, zearalenone, fulminazins, and petulin. The International Agency for Research on Cancer was categorized the mycotoxins based on their carcinogenicity potential. As you see, aflatoxin B1, B2, and aflatoxin M1 are the most carcinogenic mycotoxins based on the IARC categorization. In the following section, I will explain in detail. The most common types of aflatoxin based on the fluorescent feature are B1, B2, G1, and G2. Aflatoxins are often produced by Aspergillus flavus, Aspergillus parasiticus, and Aspergillus nomeus, mainly in warm and humid weather. Aflatoxin B1, a potent carcinogenic metabolite, is well known as the most toxic mycotoxin. Aflatoxins are mainly found in cereal and cereal-based products, nuts, oilseeds, dried fruits, spices, meat, and egg. Acute aflatoxicosis with high prevalence in developing countries occurs when a high concentration of aflatoxin is taken into the body by food consumption. In 2004, there was an outbreak of acute aflatoxicosis in Kenya, in which maize consumption with high levels of aflatoxins was the reason. Previous studies have shown that aflatoxin is an extremely strong carcinogen in all studied species, such as mice, hamster, fish, ducks, and monkeys. Symptoms are associated with reduced production parameters, such as low appetite, weight loss, decreased digestion, decreased egg or milk production, and increased susceptibility to infectious diseases. Generally, birds are more sensitive compared to mammals, and among mammals, ruminants are the most resistant. Fish is also quite susceptible to aflatoxins, and a number of studies have reported aflatoxicosis caused by trout consumption. Aflatoxins mainly target the liver. Aflatoxin M1, a hepatotoxic mycotoxin, is a hydroxylated derivative of aflatoxin B1, which exists in the milk of mammals as a consequence of aflatoxin B1 contaminated feedstuff consumption. Fumonisin is another carcinogenic metabolite. It has structural similarities to sphingosine and sphingenin, provoking the sphingolipid biosynthesis inhibition through prevention of the sphingenin N-acyl transferase as a key enzyme in the sphingolipid metabolism. Liver cancer and hepatic necrosis have been observed in mice and rats as a result of fumonisin intake. Among different foods, corn and corn-based products have shown the highest levels of contamination with fumonisin. Ochratoxin may induce mitochondrial damage, which results in oxidative degradation, lipid peroxidation, interference with oxidative phosphorylation, and inhibition of ATP synthesis. Ochratoxin is similar to the phenylalanine and therefore has an inhibitory effect on some enzymes and may lead to the inhibition of protein synthesis. Ochratoxin increases the risk of apoptosis in the cells. Ochratoxin is known as a strong nephrotoxic compound, which is often associated with urinary tract tumor in humans. In animals, the consumption of feed contaminated with ochratoxin is accompanied by an increased incidence of testicular cancer. Also, prolonged exposure to ochratoxin has been associated with nephropathy related to nephron degeneration and kidney interstitial fibrosis. Type A and B of trichotisins are separated from each other by presence or absence of a carbonyl group in their structure. These mycotoxins are common pollutants in poultry feeds. They have negative impact on the health and productivity of poultry. All of trichotisins have an epoxy in their structure, which are responsible for their toxic activity. At the cellular level, the major toxic effects of trichotisins are related to inhibition of the protein, RNA, and DNA synthesis due to interaction with the peptidyl transferase enzymes, and inhibition of mitochondrial function, causing apoptosis in animal cells. T2 toxin stimulates the suppression of the immune system in cattle by altering the concentration of A, M, and G immunoglobulins, changing the function of neutrophils and response of lymphocytes, which in turn leads to sensitivity against infectious diseases in animals. Infertility and abortion in the last trimester of pregnancy in cattle have also been associated with the consumption of feed contaminated with T2 toxin. Zearalenone is a phytoestrogenic mycotoxin produced by Fusarium species. It competitively prefers estrogen receptors. Toxicity of zearalenone is associated with reproductive and fertility disorders in certain animal species and possibly in humans. Another mycotoxin is petulin. Petulin is produced by a wide range of fungal species, including Penicillium, Aspergillus, and Bysoclamus. Penicillium expansum is well known as the most common fungal contaminants in damaged fruits. Similar to other mycotoxins, petulin is able to change the host's immune response. It affects the immune system by increasing lymphocytes and neutrophils and decreasing the immunoglobulin concentration. This toxin can also alter the function of the intestine and sometimes causes inflammation and intestinal bleeding. Patulin is able to inhibit the formation of key enzymes, such as DNA polymerase and tRNA synthetase, due to the strong tendency to bind to sulfhydryl groups. In the second part, we talk about preventive strategies. The risk of fungal contamination can be decreased by proper pre- and post-harvesting techniques. Strategies to prevent fungal contamination and mycotoxin production should be carried out on the entire food production chain, including plant growth, harvesting, storage, and distribution. Control measures must be considered before any fungal contamination or during the period of the mold invasion. Removal methods for mycotoxins is complicated, so inhibition of fungal growth is fiercely recommended and emphasized. There are some important criteria to choose a suitable method to control mycotoxins. For example, the method should be applicable and safe, shouldn't induce a significant change in the nutritional value, or shouldn't generate more toxic compounds and shouldn't release hazardous residues. Here, I'm going to talk about some important pre-harvesting strategies. The recommendations of Codex Alimentarius for prevention and reduction of mycotoxins in grains and raw materials are focused on the guidelines of good agricultural practice, good manufacturing practice, and application of HACCP system. Seeds must be free from pests and diseases prior to cultivation, and their complete health must be guaranteed. Farmers tend to select resistant varieties to aspergillus growth and aflatoxin formation. Planting date is another important management factor in controlling mycotoxins, which has an indirect impact on the production and infection of fungi spores. Planting date coincidence with spore dispersal may intensify the possibility of fungal attacks. The incidence of fungi in damaged seeds by insects has been significantly higher than the healthy seeds. Insects can act as carriers of fungal spores or create critical crop points to grow fungi and produce toxins. So, insecticide usage will probably continue as an important management tool. Insect resistant varieties are usually selected by farmers. Biotechnology experts are working on developing cereals with novel traits, including herbicide tolerance, antibiotic resistance, insect resistance, drought stress tolerance, and viral disease resistance, which are capable of reducing the risk of fungal infection. Developing healthier corn plants with high tolerance to environmental stress factors will lead to mycotoxin reduction. For example, genetically modified corn is less contaminated with mycotoxins than the conventional non-GM varieties. Post-harvesting strategies. Storage conditions are extremely important in decreasing mold growth and mycotoxin presence in foods and feeds. For example, grains must be stored at less than 15% moisture content, low temperature, low oxygen concentration, less than 1%, and increased carbon dioxide concentrations to prevent mold growth. Prolonged storage of seeds must be avoided as far as possible. Elimination of the infected plants can obviously reduce mycotoxin levels. For example, removing rotten and poor quality fruits or cutting out the contaminated parts of the fruit and then washing them can significantly reduce patulin levels up to 99% in fruit products. Most of the above strategies are effective in reducing the growth of mycotoxigenic fungi and mycotoxin production. However, once mycotoxin production occurs, contaminated food and feed must be detoxified. I divided the detoxification strategies into three parts: physical, chemical, and biological treatments. In physical treatment, for example, irradiation as a cost-effective non-thermal technology is of great interest to apply for microbial inactivation and mycotoxin degradation without any damage to the organoleptic properties or nutritional value. For example, only 60 minutes of ultraviolet radiation can decrease the acrotoxin to acceptable level in feed of poultry. Or, for example, the radiation process can reduce the aflatoxin B1 in contaminated soy seeds and peanut. It should be noted that radiated foods must be labeled and monitored according to the laws. Using adsorbents is the most common approach to reduce the mycotoxin exposure. Adsorbent materials are large molecular compounds that bind to the mycotoxins in the contaminated feed, and after that they are excreted through urine and stool, so it can reduce mycotoxin bioavailability after consumption. We have different kinds of adsorbents, for example, mineral absorbents, organic absorbents, and magnetic adsorbents. Activated carbon can reduce the amount of aflatoxins in cow's milk. Or, for example, magnetic particles have a good effect on the adsorption of petulin without toxic response in rats. I've plotted this picture for you to show the mechanism of aflatoxin B1 absorption by aluminosilicate as a famous absorbent. Mycotoxins may be connected to adsorbents by various methods, such as partition adsorption, ion exchange, or hydrogen bonding. For example, aluminosilicate with a negatively charged surface can absorb 80% of aflatoxin B1 without any adverse effects, which is added to the chicken feed to prevent aflatoxicosis. This is another absorbent, zeolite. The zeolite structure consists of a set of silicon oxide and aluminium oxide that are joined together to create a unique three-dimensional structure. Previous studies show that zeolite can protect chickens against foods contaminated with aflatoxin B1. Chemical treatment. Various chemicals, including oxidizing agents, reducing agents, acids, bases, salts, and even chlorine, have been tested for the ability to degrade mycotoxins in agricultural products. Mycotoxins are stable compounds, so no chemical action can be applied without changing the grain's nutritional value. For example, ammonia can reduce contamination, but at the same time the nutritional value would be lowered. Alkaline compounds, such as ammonia, are mainly used to remove aflatoxins from animal feed. Sodium bisulfite can destroy mycotoxins in corn and dried figs, especially aflatoxin B1. Ozonation treatment resulted in a significant reduction for mycotoxins and total fungal count. Essential oils show a high potential to reduce fungal infection and mycotoxin contamination. The activity of the natural essence of turmeric was investigated against Aspergillus flavus and aflatoxins. As a result, the inhibitory effects of turmeric essence on fungal contamination was observed in corn. Also, turmeric essence is able to inhibit the full growth of fusarium and production of zearalenone. The peppermint essence is able to prevent Aspergillus flavus from production of aflatoxin B1 in peas. Ochratoxin production can be significantly reduced by garlic and mint essences. Biological treatment. A wide range of living microorganisms, including bacteria, fungi, and yeasts, can metabolize, degrade, and transform mycotoxins. Biotransformation is based on the conversion of mycotoxins to non-toxic metabolites using biotransforming agents through acetylation or deacetylation or ring cleavage, isomerization, esterification, and etc. Lactic acid bacteria and Saccharomyces cerevisiae have shown a high degradation potential to a wide range of mycotoxins. As a conclusion, I want to say the risk of mycotoxin contamination in foodstuff exists from farm to table. The presence of mycotoxins in food and feed is inevitable worldwide. The previous studies on humans and animals have revealed a strong relationship between mycotoxin intake and acute or chronic toxicity, such as hepatotoxicity, nephrotoxicity, suppression of immune system, genotoxicity, and carcinogenicity. Complete removal of mycotoxins is so complicated. The potential risks associated with mycotoxin contaminated products can be decreased by a combination of effective pre-harvesting and post-harvesting strategies, followed by a suitable quality assurance programs. For more information, I refer you to our recent article. We highlighted the health effects of mycotoxins and prevention strategies in detail. Thank you so much for your time and your attention. If you have any question, please feel free to contact me. Goodbye and have a good time.